So here's something worth thinking about as you settle in tonight. Right now, in this moment, your eyes are doing something so quietly extraordinary that you've probably never once stopped to appreciate it. If you're lying in a dark room, your pupils have already dilated, opening wider to let in whatever faint light is available. The cells at the back of your eyes have switched over to an entirely different operating mode. A night mode, if you like, using a completely separate set of photoreceptors than the ones you were using a few hours ago in daylight. And all of this happened without you deciding to do it, without you even noticing. Your eyes just handled it. We take this for granted. Of course we do. Seeing feels effortless, like it's just happening. But what's actually going on inside that little sphere sitting in your skull is one of the most elaborate, finely tuned pieces of biological engineering that has ever existed on this planet. And I mean that literally. The human eye is not just impressive in a hand-wavy, isn't nature wonderful kind of way. It is a genuine optical instrument with a compound lens system, an adjustable aperture, an autofocusing mechanism, a self-cleaning surface, a shock-mounted housing, and a sensor array at the back that can detect a single photon of light under the right conditions. A single photon. That's the smallest unit of light that exists, and your eye can pick it up. So tonight, let's just talk about all of it. How the eye actually works, piece by piece, layer by layer. How people figured it out over centuries of careful observation and sometimes spectacularly wrong guesses. How the engineering of the eye connects to cameras, to telescopes, to fiber optics, to things you encounter every single day. There's no rush here. We'll take it slow, and if you drift off somewhere along the way, that's perfectly fine. That's kind of the point. But if you stay with me, I think you'll find that the deeper you go into how vision actually works, the more astonishing it becomes. Let's start at the very beginning. And by the beginning, I don't mean the anatomy. We'll get there. I mean the question itself. How do we see? What is sight? Because for most of human history, people got this fundamentally, completely wrong. The ancient Greeks had a lot of ideas about vision, and the most popular one for centuries was something called the emission theory. Empedocles, writing around 450 BC, proposed that the eye sent out rays, like little invisible beams, that reached out, touched objects, and brought information back. Think of it like sonar, but with light going outward from your eye. Plato liked a version of this idea too. In the Timaeus, he described a kind of gentle fire that flowed out from the eyes and merged with daylight to create the sensation of sight. And honestly, there's a certain intuitive logic to it, right? When you look at something, it does feel active. It feels like you're reaching out with your gaze. You direct your eyes at things. You aim them. So the idea that something was going outward from the eye felt natural. But here's the thing. It's completely wrong. Your eyes don't emit anything. They receive. They're passive collectors of light that's already bouncing around the world. And the person who really nailed this down, who built the argument so carefully and so thoroughly that it essentially settled the debate, was a scholar working in Cairo around the year 1000 AD. His name was Ibn al-Haytham, and he is, without exaggeration, one of the most important scientists in the entire history of optics. Some people call him the father of modern optics, and that's not an overstatement. Ibn al-Haytham, sometimes Latinized as Al-Hazan, wrote a massive 7-volume work called the Book of Optics, Kitab al-Manazir in Arabic, and in it, he systematically demolished the emission theory. His argument was elegant. He pointed out that if you stare at the sun, it hurts. It can damage your eyes. If the eye were the one sending out rays, Why would looking at a bright light cause pain? The light is clearly coming in, not going out. He also observed that when you walk from a dark room into bright sunlight, you're momentarily dazzled. Your eyes need time to adjust. Again, this only makes sense if the eye is receiving light and receiving too much of it all at once. But Ibn al-Haytham went further than just disproving the old theory. He proposed a new one, and it was remarkably close to what we understand today. He argued that every point on the surface of an illuminated object sends out light in every direction. Rays scatter off surfaces and travel in straight lines, and some of those rays enter the eye. The eye then somehow processes them into an image. He didn't have the full picture. He thought the lens was the primary sensing organ of the eye, rather than the retina, and he didn't fully understand refraction through the eye's internal structures, but his fundamental framework was right. Light comes from outside. It enters the eye. The eye processes it. Vision is reception, not emission. And here's what's really remarkable about Ibn al-Haytham. He didn't just theorize, he experimented. He built a camera obscura, a dark room with a small hole in one wall, and showed that light from outside projected an inverted image on the opposite wall. He used this to demonstrate that light travels in straight lines and that images can be formed by light passing through a small aperture. This is, in essence, the same principle your eye uses. Your eye is a camera obscura, a dark chamber with a small opening at the front and a screen at the back. He saw that connection over 1,000 years ago. Now, the camera obscura itself was known before Ibn al-Haytham. The Chinese philosopher Mozi described the phenomenon around the 4th century BC, and Aristotle noticed that sunlight passing through gaps in leaves during a partial eclipse projected little crescent shapes on the ground, each gap acting as a tiny pinhole camera. But Ibn al-Haytham was the one who really used the camera obscura as a scientific instrument, a way to study and prove things about the behavior of light. And his work, when it was translated into Latin in the 12th and 13th centuries, absolutely transformed European optics. Roger Bacon read him. Johannes Kepler read him. Without Ibn al-Haytham, the whole trajectory of optical science in Europe would have been different. So let's hold on to that image of the camera obscura for a moment, because it's going to be our anchor as we start looking at the actual anatomy of the eye. A dark chamber, a small opening, a surface at the back where the image forms. That's the basic architecture, and your eye follows it with some brilliant elaborations. Let's start from the outside and work our way in. The first thing light encounters when it reaches your eye isn't actually the pupil or the iris, it's the cornea. And the cornea is fascinating because it does most of the heavy optical lifting in your entire visual system. When people think about focusing light in the eye, they tend to think about the lens, that flexible disc sitting behind the iris. And the lens is important, absolutely. But the cornea actually provides about 2/3 of the eye's total focusing power, roughly 43 diopters out of about 60 total, if you want the numbers. The lens handles the remaining 15 to 20 or so, and most of that is for fine-tuning, adjusting focus between near and far objects. Why does the cornea do so much? It comes down to something called refraction, the bending of light as it passes from one medium into another with a different density. You've seen this 1,000 times. Put a straw in a glass of water and it looks bent at the surface. That's refraction. Light changes speed when it moves between materials of different optical densities, and when it hits the boundary at an angle, that speed change causes it to bend. Now, the biggest refraction event in your eye happens right at the front surface of the cornea, because that's where light transitions from air, which has a refractive index of essentially 1.0, into the cornea, which has a refractive index of about 1.376. That's a big jump. That's a lot of bending. And the cornea is curved, shaped like a dome, so it bends the light inward, converging it toward a focal point at the back of the eye. Think of it like a fixed lens on a camera, one that's always there, always bending light by the same amount. And here's a detail that I find quietly amazing. The cornea is transparent, completely clear, and that's actually a very unusual property for living tissue. Most of your body is opaque. Skin, muscle, bone. Light doesn't pass through any of it. So how does the cornea manage to be transparent? It comes down to its structure. The cornea is made of collagen fibers, just like a lot of other connective tissue in your body. But in the cornea, those collagen fibers are arranged in an extraordinarily precise, regular lattice, uniformly spaced, uniformly sized, laid out in parallel sheets. This regularity means that when light scatters off one fiber, the scattered waves from neighboring fibers interfere destructively in all directions except forward. The light just passes straight through. If you disrupt that regular spacing, say through swelling or injury, the cornea turns hazy. It clouds up. The engineering tolerance on that collagen lattice is incredibly tight. The cornea also has no blood vessels, none. It's one of the very few tissues in the human body that is completely avascular, and that's not an accident. Blood vessels would scatter light, creating shadows and reducing clarity. So instead, the cornea gets its oxygen and nutrients in other ways. The outer surface absorbs oxygen directly from the air, literally from the thin film of tears sitting on top of it. The inner surface gets nutrients from a fluid called the aqueous humor that fills the chamber just behind it. It's a clever workaround. No blood supply means no blood vessel shadows, means perfect transparency. And that film of tears on the surface? That matters optically too. The tear film is actually the very first refracting surface that light hits. It smooths out tiny irregularities on the corneal surface, creating a nearly perfect optical surface. When your tear film breaks up, when your eyes get dry, your vision actually degrades slightly. Things get a little blurry, a little scattered. That's why you blink. Every blink resurfaces the cornea with a fresh, smooth layer of tears. You're essentially repolishing the front element of your optical system about 15 to 20 times per minute, all day, every day, without thinking about it. So light has passed through the tear film and the cornea, and it's already been significantly bent inward, converging toward a focal point. Next, it enters the anterior chamber, a small space filled with that aqueous humor I mentioned. This is a clear, watery fluid, constantly produced and drained, and it serves a couple of purposes. It maintains the pressure inside the eye, keeping the eyeball inflated to its proper shape, and it provides nutrients to the cornea and the lens, neither of which have their own blood supply. The pressure regulation part is important, and we'll come back to it later when we talk about glaucoma, because when the drainage system for that fluid gets blocked, pressure builds up, and that pressure can damage the optic nerve. But for now, just picture this clear fluid filling the space between the cornea and the next structure the light encounters. And that next structure is the one everyone notices first when they look at someone's face. It's the part of the eye that gives you your eye color. The iris, that beautiful ring of color, brown, blue, green, hazel, gray, is actually a muscular diaphragm, and its job is pure engineering. It controls the size of the pupil, which is nothing more than a hole. The pupil isn't a structure. It isn't a thing. It's an absence, an opening in the center of the iris through which light passes. When the iris contracts, the pupil gets smaller, letting in less light. When the iris relaxes and pulls back, the pupil dilates, letting in more. This is exactly what the aperture does in a camera. Exactly. And just like a camera aperture, the size of the pupil affects two things: the amount of light reaching the sensor and the depth of field. A smaller pupil means greater depth of field. More things at different distances appear in focus simultaneously. A larger pupil means shallower depth of field. Photographers play with this all the time. Your eye does it automatically. The iris has two sets of muscle fibers to achieve this. One set is arranged in a ring, circling the pupil like a drawstring. When these contract, they squeeze the pupil smaller. The other set radiates outward like the spokes of a wheel. When these contract, they pull the iris back and the pupil opens wider. Two opposing muscle groups working in balance, controlled by your autonomic nervous system. You don't decide to do it. It just happens, responding to light levels, to emotional state, to focus distance, even to cognitive load. Your pupils actually dilate slightly when you're thinking hard about something, which is a detail that researchers have found genuinely fascinating, because it means your body is leaking information about your mental effort through your eyes without you even knowing. Psychologists like Daniel Kahneman used pupil dilation as a window into cognitive processing back in the 1960s. He'd give people math problems of increasing difficulty and watch their pupils grow as the problems got harder. The harder the problem, the wider the pupil. It's subtle. We're talking fractions of a millimeter, but it's measurable and consistent. Your eyes are literally broadcasting how hard your brain is working, and that's just one of the involuntary signals the pupil responds to. Fear dilates your pupils. Attraction dilates your pupils. Certain drugs do it too. In fact, the word belladonna, which is Italian for beautiful woman, comes from a plant extract that Renaissance women used to drop into their eyes to dilate their pupils, because wide pupils were considered attractive. They were dosing themselves with atropine, which blocks the parasympathetic signals that constrict the pupil. They didn't know the pharmacology, obviously, but they knew the effect. Wider pupils, more alluring gaze. The things people have done to their eyes throughout history would make you wince. But let's move deeper, because we've spent a good amount of time on the iris and the pupil, and there's something sitting right behind them that deserves real attention: the lens. And this is where the eye does something that no camera did for most of the history of photography. It changes shape. The lens of the eye is this remarkable little structure, roughly the size and shape of an M&M candy, maybe 9 or 10 millimeters across and about 4 millimeters thick, though those dimensions change, and that's exactly the point. It sits just behind the iris, suspended in place by a ring of incredibly fine fibers called zonules, sometimes called the zonules of Zinn, named after Johann Gottfried Zinn, a German anatomist who described them in the 1750s. These zonular fibers radiate inward from a ring of muscle called the ciliary muscle, and they hold the lens in tension, like the guy wires holding up a tent pole. And the whole system works in a way that seems almost backward until you think about it carefully. Here's how it goes. When you're looking at something far away, the moon, a distant building, a bird on a wire, your ciliary muscle is relaxed. When it's relaxed, the ring of muscle is at its widest diameter, which means the zonular fibers are pulled taut, and they pull the lens into a flatter, thinner shape. A flatter lens bends light less, which is what you need for focusing on distant objects, because those light rays are arriving nearly parallel, and don't need much bending to converge on the retina. Now, when you shift your gaze to something close, a book, your phone, the face of someone sitting across from you, the ciliary muscle contracts. When it contracts, the ring of muscle gets smaller in diameter, like tightening a drawstring. This releases the tension on the zonular fibers, and the lens, which is naturally elastic, springs into a rounder, fatter shape. A rounder lens bends light more, which is what you need to focus on nearby objects, because those light rays are diverging more strongly and need more bending to come together on the retina. This whole process is called accommodation, and it's genuinely elegant. The lens doesn't have any muscles inside it. It doesn't actively squeeze itself into shape. It's passive in a sense. It wants to be round, and the zonules are what hold it flat. So focusing on something close is actually the system letting go, releasing tension, allowing the lens to relax into its natural curvature, and focusing on something far away is the system pulling the lens taut. Think about that for a second. The resting state of the lens, its default shape, is the one optimized for close-up vision. The system has to actively work to flatten it for distance. There's something almost philosophical about that, though I won't push the metaphor too far. The person who really figured out accommodation was Hermann von Helmholtz, the German physician and physicist, in the 1850s. Helmholtz was one of those towering 19th century intellects who seemed to do groundbreaking work in whatever field he wandered into. Thermodynamics, electrodynamics, the philosophy of science, and, crucially for us, the physiology of vision. His book, The Handbook of Physiological Optics, published in stages through the 1850s and 1860s, was a monumental work. It's hard to overstate how comprehensive it was. He built instruments, designed experiments, measured the curvature of the lens during accommodation using reflections off its surfaces, a technique involving what are now called Purkinje images, named after John Evangelista Purkinje, the Czech physiologist who first described them. By carefully observing how reflections from the front and back surfaces of the lens changed when a person shifted focus from far to near, Helmholtz confirmed that the lens was physically changing shape. It wasn't moving forward and backward like a camera lens. It was reshaping itself. That was the key insight, and it settled a debate that had been going on for decades. Now, the lens itself is a beautiful piece of biological engineering. It's made up of long, thin, transparent cells called lens fibers, packed together in concentric layers like the layers of an onion. New fibers are added to the outside throughout your entire life, while the older fibers in the center get compressed and compacted into what's called the nucleus of the lens. The outer, softer region is the cortex. And here's a critical detail. The cells in the center of the lens have no nucleus, no organelles. They've essentially been stripped down to nothing but transparent, densely packed crystalline proteins. The cell literally sacrifices its own internal machinery to achieve optical clarity. If those organelles were still in there, they'd scatter light. So the lens cells do something almost unprecedented in biology. They destroy their own insides to become perfectly transparent. It's a one-way trip. Those cells can never repair themselves, can never divide again. They're locked in place for the rest of your life. And this is where the story gets a little bittersweet, because the lens is the part of the eye that ages most noticeably. Remember how accommodation depends on the lens being elastic, springing into that rounder shape when the zonules release tension? Well, as you get older, the lens gradually stiffens. New layers keep being added to the outside, compressing the interior, and the crystalline proteins in the center slowly lose their flexibility. By the time most people hit their early to mid-40s, the lens has become rigid enough that it can't bulge into that round shape as effectively anymore. Close-up focus gets harder. You start holding your phone a little farther away. You find yourself needing more light to read. This condition is called presbyopia, from the Greek for old eye, and it's essentially universal. It doesn't matter how good your vision was when you were 20. It doesn't matter if you're nearsighted or farsighted or had perfect vision your whole life. Presbyopia comes for nearly everyone because it's not a disease. It's a consequence of the lens continuing to grow and stiffen over decades. It's the one optical change in the eye that's truly age-dependent and almost impossible to prevent. There's something humbling about that. The same process that keeps the lens transparent, adding new layers, compacting old ones, is the process that eventually robs it of flexibility. The system's greatest strength becomes its limitation over time. And if the lens stiffens enough, or if those crystalline proteins start to clump and aggregate, you get a cataract. The lens goes cloudy. Light scatters instead of passing through cleanly, and vision gets hazy, washed out, like looking through a fogged-up window. Cataracts are the leading cause of blindness worldwide, and they've been recognized for thousands of years. The ancient Indians, particularly a surgeon named Sushruta, described a technique for dealing with cataracts around the 6th century BC, or possibly earlier, a procedure called couching, where a sharp instrument was used to push the clouded lens down and out of the line of sight into the vitreous cavity of the eye. It didn't remove the cataract, it just shoved it out of the way. The patient could see again. Sort of, everything was extremely blurry, because now there was no lens at all to help focus light, but it was better than the milky blindness of a dense cataract. Couching was practiced across the ancient world, from India to Greece to the Islamic world, for centuries. It's remarkable when you think about it. People were performing intraocular surgery thousands of years before. Germ theory, before anesthesia, before anyone truly understood what the lens did optically. The courage of both the surgeons and the patients is hard to fathom. Modern cataract surgery, of course, is a completely different thing. Today, the clouded lens is broken up, usually with ultrasound energy in a process called phacoemulsification, and the fragments are suctioned out through a tiny incision. Then an artificial intraocular lens, a little plastic or silicone lens, is folded up, inserted through that same small incision, and unfolded inside the eye, sitting right where the natural lens used to be. It's one of the most commonly performed surgeries in the world, and it takes maybe 15 or 20 minutes. The engineering of those artificial lenses has gotten incredibly sophisticated. There are now multifocal intraocular lenses that try to give patients both distance and near vision, mimicking some of what natural accommodation used to provide. They're not perfect, but they're a remarkable piece of biomedical engineering. But let's keep moving inward, because we've passed through the cornea, the aqueous humor, the iris and pupil, and the lens. And now the light that's made it through all of those structures enters the largest chamber of the eye, the vitreous cavity, filled with the vitreous humor. This is the big, gel-filled space that makes up about 80% of the eye's volume. The vitreous is mostly water, around 99%, but it's given structure by a sparse network of collagen fibers and hyaluronic acid molecules. It's like a very, very dilute gel, almost like a biological version of clear gelatin. And its job, in engineering terms, is partly structural. It maintains the shape of the eyeball, keeps the retina pressed against the back wall, and partly optical. It's a transparent medium that light passes through on its way to the retina. The vitreous is also where floaters come from, those little drifting specks and threads you sometimes see when you look at a bright sky or a white wall. Those are tiny clumps of collagen fibers within the vitreous, casting shadows on the retina. They're usually harmless, just a consequence of the vitreous gel slowly liquefying as you age. Another one of those gradual changes the eye undergoes over a lifetime. But occasionally, if the vitreous pulls away from the retina too abruptly, a process called posterior vitreous detachment, it can tug on the retina and cause flashes of light, or in worse cases, actually tear the retina, which is a medical emergency. The retina, as we're about to see, is not something you want damaged, because it's where the real magic happens. Everything up to this point, the cornea, the lens, the iris, all of it, has been about getting light to the right place in the right condition. The retina is where light stops being light and starts becoming something else entirely. A signal, an electrical chemical whisper that your brain can understand. And that transformation, light into neural code, is one of the most elegant pieces of biological engineering you'll ever encounter. So let's talk about the retina. And the first thing to appreciate is that the retina is not just a screen. It's not like the film in a camera or the sensor in your phone. It's actually a piece of your brain. Embryologically, the retina develops from the same tissue that becomes your central nervous system. During fetal development, two little pouches bulge outward from the developing brain, and those pouches eventually become the retinas of your two eyes. So when you look someone in the eye, you are, in a very real sense, looking at a piece of their brain, the only part of the central nervous system that's exposed to the outside world. And that fact alone should tell you something about how complex this tissue is going to be. The retina is thin, remarkably thin. We're talking about a tissue that's roughly 1/4 millimeter thick in most places, thinner than a sheet of plastic wrap. And yet, packed into that sliver of tissue are over 100 million photoreceptor cells, plus several layers of neurons that begin processing visual information before it even leaves the eye. The retina doesn't just detect light, it starts interpreting it. It performs edge detection, contrast enhancement, motion sensing, computations that engineers working on machine vision have spent decades trying to replicate in silicon. And the retina does it all in a wet, warm, biological substrate, thinner than a credit card. Now, here's something that catches people off guard when they first learn about retinal anatomy. The retina is built from an engineering standpoint, seemingly backwards. The photoreceptors, the cells that actually detect light, are at the back of the retina, farthest from the incoming light. In front of them, between the light and the photoreceptors, sit multiple layers of neurons, blood vessels, and supporting cells. Light has to pass through all of that stuff before it reaches the cells that are designed to catch it. It's as if you built a camera and then put the wiring and circuit boards in front of the sensor. Why on earth would evolution do this? The answer comes down to metabolism. Photoreceptors are among the most metabolically demanding cells in your entire body. They consume oxygen and nutrients at a ferocious rate. Some estimates suggest that the photoreceptors in your retina, pound for pound, burn through more energy than almost any other tissue you have. And to feed that appetite, they need to be right next to their blood supply, which comes from a layer of tissue behind the retina called the retinal pigment epithelium, or RPE. The RPE sits against a dense bed of blood vessels called the choroid, and together they form a kind of service infrastructure, delivering nutrients, recycling waste products, and doing critical chemical maintenance that keeps the photoreceptors functioning. So the photoreceptors face backward toward their supply lines, and light has to thread its way through the neural layers in front to reach them. It's a design compromise, and honestly, it works astonishingly well because those neural layers in front are mostly transparent. Light passes through them with very little loss. But the compromise does have consequences. It's part of the reason retinal detachment is possible and so dangerous, because the photoreceptors can peel away from the RPE that sustains them. But let's get to the photoreceptors themselves, because this is where the story gets truly beautiful. You have two main types, rods and cones, and they're named, simply enough, for their shapes. Rods are elongated, cylindrical, cones are shorter, tapered, and they do fundamentally different jobs. Rods are your low-light specialists. You have roughly 120 million of them in each eye, and they are extraordinarily sensitive. A single rod cell can respond to a single photon of light, one quantum of electromagnetic energy. Think about that for a second. One photon. That's the smallest unit of light that exists, and your eye has cells that can detect it individually. The physicist Selig Hecht and his colleagues demonstrated this in a famous series of experiments in the 1940s at Columbia University. They sat subjects in complete darkness for long periods to fully dark-adapt their eyes, then flashed incredibly dim lights and asked whether the subjects could see them. By carefully controlling the intensity and doing statistical analysis, Hecht's team concluded that human vision at its absolute threshold requires something like 5 to 7 photons being absorbed by rod cells within a short time window. Not 5 to 7 photons hitting the eye. Many more than that hit the cornea. But 5 to 7 actually being captured by rod photoreceptors. And since each rod can respond to a single photon, that means the limiting factor isn't the sensitivity of individual cells. It's the brain's requirement for a quorum, a minimum number of simultaneous signals to distinguish a real flash from random neural noise. The engineering logic there is noise reduction. Your rods are so sensitive that they occasionally fire spontaneously, producing false signals. So your visual system demands a consensus, multiple rods reporting in at roughly the same time, before it believes something is really out there in the dark. It's the same principle behind requiring multiple independent sensors to agree before triggering an alarm system. Cones, on the other hand, are your daylight and color sensors. You have far fewer of them, only about 6 to 7 million per eye, and they're much less sensitive than rods. They need a lot more light to activate. But they give you two things rods can't: color vision and high spatial resolution. And the way they provide color vision is one of those ideas that took centuries to work out. Thomas Young, the same polymath who demonstrated the wave nature of light with his famous double-slit experiment in 1801, proposed that human color vision could be explained by just 3 types of receptors, each sensitive to a different range of wavelengths. Young's idea was remarkably ahead of its time. He didn't know about cone cells specifically, but he reasoned, purely from the physics and the observed behavior of color mixing, that 3 channels would be sufficient to produce the full range of colors we perceive. Hermann von Helmholtz. And yes, he keeps showing up in this story, because the man was everywhere in 19th century vision science. Helmholtz picked up Young's idea decades later, refined it, provided more experimental support, and the framework became known as the Young-Helmholtz trichromatic theory, and it turned out to be fundamentally correct. We now know that you have 3 types of cone cells, each containing a slightly different version of a light-sensitive protein called an opsin. One type, called S cones, is most sensitive to short wavelengths, what you'd perceive as blue-violet light, peaking around 420 nanometers. Another type, M cones, responds best to medium wavelengths, peaking around 530 nanometers, green. And the third type, L cones, is tuned to longer wavelengths, peaking around 560 nanometers, which is actually more yellow-green than pure red, though we call them the red cones. Every color you've ever seen, every sunset, every painting, every neon sign, your brain constructed from the relative activation levels of just these three cone types. When all three fire roughly equally, you see white. When L cones fire much more than the others, you see red. When S cones dominate, blue. Your brain is doing a kind of triangulation, comparing the ratios of signals from three channels and computing a color percept from those ratios. It's essentially the same principle used in every color screen you've ever looked at. Your phone, your TV, your computer monitor. They all use three color primaries, red, green, and blue, mixed in different intensities to create every color. The engineers who designed those screens were directly exploiting the trichromatic architecture of your retina. But here's the thing. The story of color vision doesn't end with Young and Helmholtz, because trichromatic theory, while correct at the receptor level, couldn't explain everything people observed about color perception. There were phenomena, like why you never see a reddish green or a yellowish blue, or why staring at a red patch and then looking at a white wall produces a green afterimage, that three-channel mixing alone couldn't account for. And this is where a rival theory enters the picture, proposed by Ewald Hering, a German physiologist working in the late 1800s. Hering argued that color vision was organized into opponent pairs, red versus green, blue versus yellow, and black versus white. Colors in each pair were antagonistic. The visual system processed them as opposites. You could have reddish yellow or greenish blue, but never reddish green, because those two were on opposite ends of the same channel. For decades, the Young Helmholtz camp and the Hering camp were at odds. It was one of those scientific rivalries where both sides had compelling evidence and neither could fully account for the other's observations. And the resolution, when it finally came in the mid-20th century, was wonderfully satisfying. Because it turned out both theories were right, just at different stages of processing. The cones in your retina work exactly as Young and Helmholtz described. Three types, trichromatic coding. But the neurons that the cones connect to, in the retina itself, and then further along in the brain, reorganize those three signals into opponent channels, exactly as Hering described. So, trichromatic theory describes what happens at the photoreceptor level, and opponent process theory describes what happens at the neural processing level. The two theories weren't competing explanations. They were complementary descriptions of different stages in the same pipeline. And that resolution, that moment of realizing two seemingly contradictory ideas were both true, is one of the most satisfying things in the history of vision science. Now, let's go deeper into how a photoreceptor actually converts light into a signal, because the molecular mechanism, called phototransduction, is genuinely one of the most remarkable cascades in all of biology. Inside each rod and cone, in the outer segment of the cell, there are stacks of membranous discs, like a roll of coins. Embedded in those disc membranes are millions of molecules of photopigment. In rods, the photopigment is called rhodopsin. And rhodopsin is a protein, the opsin part, cradling a small molecule called retinal, which is derived from vitamin A. This, by the way, is why vitamin A deficiency causes night blindness. Without enough vitamin A, you can't make enough retinal, and without retinal, your rods can't function properly. Here's how it works. Retinal normally sits in a bent configuration called 11-cis retinal, nestled snugly inside the opsin protein. When a photon of the right wavelength strikes this molecule, it does something incredibly fast and incredibly specific. It causes the retinal to straighten out, snapping from the bent 11-cis form to a straight all-trans form. This shape change happens in about 200 femtoseconds. A femtosecond is a millionth of a billionth of a second. It's one of the fastest known chemical reactions in nature, and that one tiny molecular twitch, one molecule changing shape because one photon hit it, triggers everything that follows. The straightened retinal forces the opsin protein to change shape too, and the activated opsin, now called meterhodopsin 2, becomes an enzyme that starts a biochemical amplification cascade. One activated rhodopsin molecule activates hundreds of molecules of a protein called transducin. Each transducin activates an enzyme called phosphodiesterase. Each phosphodiesterase molecule chews through thousands of molecules of a signaling chemical called cyclic GMP. And it's cyclic GMP that holds open ion channels in the photoreceptor's membrane. As cyclic GMP levels plummet, those channels slam shut. The flow of sodium and calcium ions into the cell drops, and the cell's electrical voltage changes. It hyperpolarizes, becoming more negative inside. That voltage change is the signal. That's the moment light becomes electricity. And notice the amplification at each step. One photon activates one rhodopsin. One rhodopsin activates hundreds of transducins. Each transducin-activated phosphodiesterase destroys thousands of cyclic GMP molecules. So a single photon can ultimately close hundreds of ion channels and produce a measurable electrical change in the cell. The gain of this cascade is enormous, and it's precisely this amplification that gives rods their astonishing single-photon sensitivity. But here's what's equally impressive. The system has to turn itself off. If rhodopsin just stayed activated, the cell would be stuck, unable to respond to the next photon. So there's an entire shutdown mechanism. A protein called rhodopsin kinase tags the activated rhodopsin, and then another protein called arrestin caps it, silencing it. The all-trans retinal is recycled back to 11-cis retinal through a series of enzymatic steps, many of which happen in the retinal pigment epithelium, that surface layer behind the photoreceptors we talked about. The whole cycle of activation, amplification, shutdown, and recycling takes a fraction of a second, and it repeats continuously, millions of times, across millions of photoreceptors, every moment your eyes are open. Now, all of this molecular machinery produces a graded electrical signal in the photoreceptor, but that signal still has to get from the photoreceptor to the brain, and the retina doesn't just pass it along unchanged. Between the photoreceptors and the ganglion cells whose axons form the optic nerve, there are layers of interneurons, bipolar cells, horizontal cells, amacrine cells, and these neurons are already doing computation. They're comparing signals from neighboring photoreceptors, enhancing edges, adjusting for overall brightness levels, detecting motion. The retina is performing image processing before the signal ever leaves the eye. And the way it does that, the specific circuitry involved, starts to get into some really fascinating territory, because it reveals how your visual system solves problems that even the best camera engineers are still struggling with today. Think about one of the most basic challenges in seeing, figuring out where one object ends and another begins. You look at a coffee mug sitting on a desk, and it seems so obvious. There's the mug, there's the desk, there's the wall behind it. But from the retina's perspective, all it's receiving is a wash of photons hitting millions of photoreceptors, each one reporting its own little patch of light intensity. There are no labels, there are no outlines, just a sea of signals. So how does your visual system start carving that sea into meaningful shapes? It starts right there in the retina with those interneurons, and the key trick is something called lateral inhibition. Here's how it works. When a photoreceptor gets stimulated by light, it doesn't just send its signal straight forward to a bipolar cell. It also communicates sideways through horizontal cells to its neighbors. And the message it sends sideways is essentially inhibitory. It's dampening the signal of the photoreceptors around it. Now think about what that means at the boundary between a bright area and a dark area. The photoreceptors in the bright zone are firing strongly, and they're inhibiting their neighbors. The photoreceptors in the dark zone are firing weakly, so they're barely inhibiting anything. Right at the edge, something interesting happens. The bright side photoreceptor near the boundary is being inhibited by its bright neighbors on one side, but not much by its dark neighbors on the other side, so its signal stays relatively strong. Meanwhile, the dark side photoreceptor near the boundary is being inhibited by its bright neighbor across the edge, but getting very little excitation of its own. So its signal gets pushed even darker. The net effect is that the contrast at the edge gets exaggerated. The bright side of the boundary looks brighter than it really is, and the dark side looks darker. The retina is sharpening edges before the brain ever gets involved. This is something you can actually experience yourself. There's a classic visual illusion called Mach bands, named after the physicist Ernst Mach. Yes, the same Mach as in Mach numbers and the speed of sound. Back in the 1860s, Mach noticed that when you look at a gradient that goes from light to dark, you perceive a bright band right at the light edge and a dark band right at the dark edge, even though the actual luminance is perfectly smooth. Those phantom bands aren't in the image. They're created by your retina's lateral inhibition circuitry. Mach figured this out over 150 years ago, and it was one of the earliest insights into how neural processing shapes perception, how what you see isn't just what's there, but what your nervous system constructs. The horizontal cells handle a lot of this lateral communication, but the amacrine cells are doing something different and arguably even more interesting. There are at least 30 distinct types of amacrine cells in the human retina, maybe more, and they're involved in everything from motion detection to adaptation to changing light levels. Some amacrine cells are specifically tuned to detect the direction of movement. They respond strongly when something moves across the retina in one direction, but barely respond when it moves the other way. This is directional selectivity, and it's computed locally, right there in the retina, by the specific wiring pattern between photoreceptors, bipolar cells, and these specialized amacrine cells. You don't need the brain's visual cortex to detect motion. The retina is already on it. And then there are the bipolar cells, which are the main vertical pathway, the pipeline from photoreceptors to ganglion cells. But even here, it's not a simple relay. Bipolar cells come in two fundamental flavors: on bipolar cells and off bipolar cells. On bipolar cells are excited when light increases in their receptive field. Off bipolar cells are excited when light decreases. So right at this stage, the retina is splitting the visual signal into two parallel channels, one for brightness and one for darkness. This is a profoundly clever design choice. Instead of having one channel try to encode the full range from very dark to very bright, you split the job. One channel specializes in reporting where things are getting lighter, and the other specializes in where things are getting darker. It's more efficient, and it turns out to be a strategy that gets used over and over again throughout the visual system. All of these processed signals converge on the ganglion cells, and this is where things really come together, because the ganglion cells are the output neurons of the retina. Their axons are the fibers that bundle together to form the optic nerve. Everything the brain knows about what the eye is seeing comes through these cells. There are roughly 1 to 1.2 million ganglion cells in each human retina, which means the retina is compressing the output of over 100 million photoreceptors down to about 1 million channels. That's a compression ratio of roughly 100 to 1. And it's not just throwing away information. It's extracting the most important features and encoding them efficiently. Now, ganglion cells have what's called a center surround receptive field, and this is one of the most important concepts in visual neuroscience. The idea was worked out in detail by Stephen Kuffler in the early 1950s, working with cat retinas at Johns Hopkins. What Kuffler found was that each ganglion cell responds to a small circular area of the visual field, but the response isn't uniform across that area. There's a center region and a surrounding ring, and they have opposite effects. An ON center ganglion cell, for example, fires more when light hits the center of its receptive field, but fires less when light hits the surround. An off-center cell does the opposite. It fires when the center goes dark. If you flood the entire receptive field with uniform light, the center and surround effects roughly cancel each other out, and the cell barely responds. Think about what that means. These cells are essentially uninterested in uniform illumination. They don't care about absolute brightness. What they care about is contrast, differences between the center and the surround. They're edge detectors and spot detectors. They're encoding the structure of the image, not its overall brightness. This was a huge insight. Kuffler's work, published in 1953, fundamentally changed how neuroscientists thought about sensory processing. It showed that the nervous system isn't passively recording the world like a camera sensor. It's actively selecting for the most informative features. And this principle, that neurons respond to specific features rather than raw intensity, became the foundation for everything that David Hubel and Torsten Wiesel would later discover about the visual cortex, work that earned them the Nobel Prize in 1981. But we'll get there. The point for now is that the retina's ganglion cells are already sophisticated feature detectors, and they're not all the same. There are several major types of ganglion cells, and each type forms its own parallel information channel to the brain. The two most important types in primates are called midget cells and parasol cells, though they also go by their anatomical names. Parvocellular and magnocellular, or P cells and M cells, named for the layers of the brain relay station where their axons terminate. Midget ganglion cells are small, with tiny receptive fields, and they're concentrated heavily in the central retina, especially the fovea. They're the high-resolution channel. They carry detailed information about shape, fine texture, and color. In the fovea, a single midget ganglion cell may receive input from just one cone, giving you that incredible point-by-point acuity. Parasol cells are larger, with bigger receptive fields, and they respond best to changes, movement, flicker, rapid shifts and contrast. They're your motion and temporal change channel. They respond faster than midget cells, but with less spatial detail. So right here, before the signal has even left the eye, the retina has already split vision into parallel streams, one optimized for detailed, colorful, high-resolution seeing, and another optimized for fast, motion-sensitive, broad-stroke seeing. This is not something the brain imposes later. The retina builds it in from the start. All of these ganglion cell axons, roughly 1 million of them, converge toward one spot on the retina, a point slightly nasal to the fovea, and they bundle together and punch through the retinal layers to form the optic nerve. And here's the thing about that exit point. There are no photoreceptors there. None. It's just a hole where the nerve fibers leave and blood vessels enter. This is your blind spot, and you genuinely have one in each eye. It's not tiny either. It corresponds to a gap in your visual field roughly the size of about 10 full moons clustered together. You don't normally notice it because your brain fills in the missing information using context from the surrounding area, and because your two eyes' blind spots don't overlap, so one eye covers for the other. But it's there. You can find it with simple tests. Close one eye, stare at a fixed point, and move a small object slowly until it vanishes. It just disappears. Your brain doesn't show you a black hole or a blank patch. It just quietly paints over the gap with whatever pattern surrounds it, which, if you think about it, is both remarkable and slightly unsettling, because it means your brain is routinely editing your visual experience without telling you. The optic nerve itself is a cable of about 1 million axons, and it's technically not a peripheral nerve at all. It's a tract of the central nervous system, an extension of the brain, wrapped in the same type of myelin sheaths and meninges that protect brain tissue. This matters clinically because it means the optic nerve doesn't regenerate the way peripheral nerves can. Damage to it, from glaucoma, from trauma, from multiple sclerosis, is generally permanent, at least with current medicine. Once the optic nerve exits the back of the eye, it travels about 5 centimeters back into the skull, and then something remarkable happens. The two optic nerves from your two eyes meet at a structure called the optic chiasm, right above the pituitary gland at the base of the brain. And here, roughly half the fibers from each eye cross over to the opposite side. But it's not random. It's beautifully organized. The fibers from the nasal half of each retina, the half closer to your nose, cross over. The fibers from the temporal half, the half closer to your temple, stay on the same side. The result is that after the chiasm, the right side of the brain receives information from the left half of the visual field of both eyes, and the left side receives information from the right half. Both eyes' views of the same region of space are brought together on the same side of the brain, which is exactly what you need for binocular vision, for depth perception, for comparing the slightly different images from your two eyes, to calculate how far away things are. This arrangement was worked out anatomically over centuries, but the functional significance really became clear through clinical neurology. Doctors noticed that specific types of brain injuries produced very specific patterns of vision loss. A stroke affecting the right visual cortex would knock out the left visual field in both eyes. A tumor pressing on the middle of the optic chiasm, often a pituitary tumor, would destroy the crossing fibers and produce tunnel vision, wiping out peripheral vision on both sides while leaving central vision intact. These patterns were like a wiring diagram read backward from the symptoms, and they taught neurologists an enormous amount about how the visual pathways are organized long before modern brain imaging existed. After the chiasm, the fibers continue as the optic tracts, and most of them, about 90%, terminate in a structure deep in the brain called the lateral geniculate nucleus, or LGN, which sits in the thalamus. The LGN is often described as a relay station, and in a sense it is. It's the last stop before the visual cortex. But calling it just a relay sells it short. The LGN has 6 distinct layers, and they're organized with exquisite precision. The bottom 2 layers, the magnocellular layers, receive input from those parasol ganglion cells, the motion and change channel. The top 4 layers, the parvocellular layers, receive from the midget cells, the detail and color channel, and each layer receives input from only one eye, alternating left eye, right eye, left eye, right eye. The information from the 2 eyes is kept segregated here, laid out in neat parallel sheets, waiting to be combined in the cortex. The LGN also receives massive feedback from the visual cortex. In fact, more fibers come back down from the cortex to the LGN than go up from the LGN to the cortex, which tells you that the brain isn't just passively receiving visual data, it's actively modulating what gets through. The cortex is essentially telling the LGN what to pay attention to, sharpening some signals, suppressing others. Attention, expectation, context. All of these higher-level factors are already influencing the visual signal before it reaches the cortex for conscious processing. Your brain is, in a very real sense, choosing what to see. From the LGN, the fibers fan out in a broad sheet called the optic radiations, sweeping through the white matter of the brain toward the back of the head, toward the primary visual cortex, also known as V1, sitting in the occipital lobe. And this is where the story of visual processing takes its most dramatic turn, because what Hubel and Wiesel found there in the late 1950s and 1960s completely changed the way we understand the brain itself. David Hubel and Torsten Wiesel, working at Johns Hopkins and later at Harvard, did something beautifully simple. They inserted tiny electrodes into the visual cortex of anesthetized cats and tried to figure out what made individual neurons fire. And at first, they struggled. They'd shine spots of light on a screen in front of the cat's eyes, the same kind of spots that had worked so well for Kuffler when he mapped those center surround receptive fields in the retina and LGN. But the cortical neurons just didn't care. The cells sat there, mostly silent, and Hubel and Wiesel couldn't figure out what they wanted. The breakthrough, as Hubel later described it, came partly by accident. They were using glass slides with dark spots on them, projecting these spots onto a screen, and as they slid one of the slides into the projector, the edge of the slide itself cast a faint shadow, a straight line moving across the screen, and suddenly, the neuron they were recording from went wild. It fired like crazy, not for the spot, for the edge, for the oriented line. That was the moment. That was when they realized that neurons in the primary visual cortex aren't looking for spots of light or darkness. They're looking for edges, lines, bars, features with a specific orientation. A particular neuron might fire vigorously when it detects a line tilted at, say, 45 degrees, and barely respond at all to a horizontal or vertical line. The neuron next to it might prefer a line at 50 degrees, the one after that, 55, and so on. Think about what that means. The retina sends the cortex a map of light and dark, organized by contrast. The LGN refines and gates that signal. And then V1 takes all of that raw contrast information and starts extracting geometry. It's pulling out the building blocks of shape. Every edge of every object you've ever looked at, the rim of a coffee cup, the line where a wall meets the ceiling, the curve of someone's jaw. Your visual cortex is detecting those edges, one tiny oriented segment at a time, using neurons tuned to specific angles in specific locations of your visual field. Hubel and Wiesel classified these neurons into types. The simplest ones they called, straightforwardly enough, simple cells. A simple cell responds to an edge or a bar of light at a particular orientation in a particular location. It has distinct excitatory and inhibitory zones, almost like the center surround fields of the retina, but stretched out into elongated strips. You can imagine it as a neuron that's wired to receive input from a row of LGN cells whose circular receptive fields line up. So instead of responding to a spot, it responds to a line. It's a gorgeous piece of neural engineering, building complexity from simplicity, one layer at a time. Then they found complex cells. These also respond to oriented edges, but they're less picky about exactly where in their receptive field the edge falls. A complex cell might fire for a vertical edge anywhere within a certain region, and it often responds especially well to edges that are moving. So now you've got neurons that don't just detect that there's a vertical line. They detect a vertical line that's sweeping leftward. And then there are end-stopped cells, sometimes called hypercomplex cells, which respond to edges of a specific length. They fire for a short bar, but stop firing if the bar extends too far. These are the neurons that help you detect corners, endpoints, the places where one edge terminates and another begins. Hubel and Wiesel also discovered something remarkable about how these neurons are organized physically within the cortex. V1 isn't just a random jumble of edge detectors, it's arranged in columns, vertical columns of neurons running perpendicular to the cortical surface. All the neurons in a single column prefer roughly the same orientation. Move your electrode sideways across the cortex, and the preferred orientation shifts systematically. 10 degrees, 20 degrees, 30 degrees, rotating smoothly through a full 180 degrees and then starting over. They called these orientation columns, and layered on top of that, there are ocular dominance columns, alternating stripes where neurons respond preferentially to input from the left eye or the right eye. The two eyes, which were kept segregated in the LGN, are finally being brought together here in V1, but in an organized, interleaved pattern. So picture the surface of V1 as a kind of quilt. Each patch of cortex handles a small piece of the visual field. Within that patch, you have a full set of orientation columns covering all angles, and a pair of ocular dominance columns, one for each eye. Hubel and Wiesel called this repeating unit a hypercolumn. One hypercolumn contains everything the cortex needs to analyze one small region of visual space, all orientations, both eyes. And these hypercolumns tile the entire surface of V1, covering your whole visual field. It's modular, it's systematic, it's almost crystalline in its regularity. And for mapping this architecture, Hubel and Wiesel shared the Nobel Prize in Physiology or Medicine in 1981. But here's where their work had consequences far beyond basic science, and this is a part of the story that really matters. They also studied what happens when you disrupt visual input during early development. In a series of experiments in the 1960s, they sewed shut one eyelid of a kitten during the first few weeks of life, the critical period, and then opened it later. What they found was devastating in its clarity. The eye itself was fine. The retina worked, the optic nerve conducted signals, but the visual cortex had essentially abandoned that eye. The ocular dominance columns that should have received input from the deprived eye had shrunk, while the columns for the open eye had expanded to take over the territory. The kitten was functionally blind in the deprived eye, not because of any problem with the eye, but because the brain had never learned to use it. This discovery transformed our understanding of brain development and had immediate medical implications. It explained why children with strabismus, crossed eyes, or with cataracts present from birth need to be treated early. If you wait too long, the brain's visual cortex will reorganize itself in ways that can't easily be reversed. The critical period in humans extends roughly through the first several years of life, with the most intense plasticity in the first year or two. This is why pediatric ophthalmologists are so insistent about early screening. A child who has a dense cataract in one eye doesn't just need that cataract removed. They need it removed soon, or the cortical real estate for that eye will be permanently reassigned. The engineering of the eye might be perfect, but if the brain's wiring doesn't develop properly during that narrow window, vision is lost at a level no surgery on the eye can fix. Now, V1 is just the beginning of cortical visual processing, and this is something that can be hard to wrap your head around. V1 is enormous by cortical standards. It's the single largest area in the human cortex, and it contains on the order of 200 million neurons, but it's only the first stage. From V1, visual information flows forward into a cascade of higher visual areas, V2, V3, V4, V5, and beyond, each one extracting increasingly abstract features from the image. And here's where one of the most elegant organizational principles in neuroscience comes in. Starting from V1, visual processing splits into two major streams, two parallel pathways that handle fundamentally different questions about what you're seeing. The ventral stream flows downward and forward along the bottom of the brain into the temporal lobe. This is sometimes called the what pathway because it's specialized for object recognition, identifying what something is: color, texture, shape, faces, letters, the difference between a dog and a cat. The dorsal stream flows upward and forward into the parietal lobe. This is the where or how pathway. It handles spatial relationships, motion, depth, and the information you need to interact physically with objects. Where is that coffee cup relative to my hand? How fast is that car approaching? How do I shape my fingers to grip this doorknob? This two-stream model was proposed by Mortimer Mishkin and Leslie Ungerleider at the National Institutes of Health in the early 1980s, and while it's been refined and complicated since then, the basic framework has held up remarkably well. And the most compelling evidence for it comes from clinical neurology, from patients with damage to one stream but not the other. Consider a condition called visual agnosia, which can result from damage to the ventral stream. A patient with visual agnosia can see perfectly well in many respects. Their eyes work. Their visual acuity might be normal. They can navigate a room without bumping into furniture. They can catch a ball thrown to them. But they cannot recognize objects. They might look at a fork and have no idea what it is. They can describe it. It's a metal thing with prongs, but the visual form doesn't connect to meaning. The most famous case study is probably the man described by Oliver Sacks in The Man Who Mistook His Wife for a Hat. A musician with a degenerative condition affecting his ventral stream who literally could not distinguish his wife's face from a hat. His dorsal stream was intact, so he could move through the world, reach for things, judge distances. But the what channel was broken. Now flip it. damage to the dorsal stream produces a completely different syndrome. A patient might recognize objects perfectly. They know that's a cup, that's a pen, that's a face. But they can't reach accurately for the cup. They misjudge distances. They can't perceive motion properly. In the most striking cases, a condition called achinotopsia, damage to area V5, also called MT for the middle temporal area, eliminates the perception of motion entirely. The world appears as a series of frozen snapshots. A patient described by the neurologist Joseph Ziel in the 1980s reported that pouring coffee was terrifying because the liquid appeared frozen in midair, and then suddenly the cup was overflowing. Cars didn't appear to move. They were just suddenly in a different place. Imagine that for a moment. Everything else about vision is intact. Color, shape, detail, recognition. But motion, something so fundamental that you never even think about it, is simply gone. These cases reveal something profound about how vision actually works. It's not one thing. It's dozens of parallel processes happening simultaneously, each handled by specialized neural machinery, and your conscious experience of seeing this seamless, unified visual world that feels so effortless and whole is a construction. It's assembled from all these separate streams, stitched together so smoothly that you never notice the seams, until one piece breaks, and then you realize that what felt like a single experience was actually an orchestra of processes playing in concert. And speaking of that seamless construction, there's another piece of the puzzle that's worth lingering on, because it reveals just how much your brain is doing behind the scenes that you're completely unaware of. Let's talk about color, not the physics of color, which is about wavelengths of light, and not the photoreceptor side, which we've already covered with the 3 cone types. Let's talk about what happens after that. Because the way your brain constructs color from those 3 cone signals is far stranger and more creative than you might expect. Your brain doesn't just read out the wavelength of light hitting your retina and assign a color to it. If it did, the colors of objects would shift wildly every time the lighting changed, stepping from fluorescent office light into warm afternoon sunlight would make everything look completely different. But it doesn't, does it? A red apple looks red under fluorescent light, under sunlight, under overcast skies, even under fairly extreme color shifts. This phenomenon is called color constancy, and it's one of the most impressive computational feats your visual system performs. Your brain is essentially estimating the color of the illumination, the overall tint of the light source, and then subtracting it out so that you perceive the intrinsic color of the object rather than the raw wavelength reaching your eye. Edwin Land, the inventor of the Polaroid camera, did some of the most striking demonstrations of this in the 1950s and 1960s with what he called his Retinex theory, showing that you could create a full range of perceived colors using only 2 wavelengths of light if you set up the scene correctly. The brain was filling in the rest, constructing color from context and comparison rather than from absolute wavelength measurements. And that process, that active construction, happens in the ventral stream, particularly in an area called V4, where neurons respond not to raw wavelength, but to perceived color, to the color your brain has decided something actually is, after all that contextual computation has been done. And that distinction between the wavelength that physically hits your retina and the color your brain decides you're seeing is one of the most profound things about vision, because it means color, as you experience it, is a construction. It's an opinion your brain forms after weighing evidence. Let's stay with V4 for a moment, because the story of how we know what it does is itself pretty fascinating. In the late 1970s and into the 1980s, a neuroscientist named Samir Zeki at University College London was doing careful recordings in monkey brains, mapping out the visual areas beyond V1, and he found that neurons in V4 had this remarkable property. You could shine a patch of light on a monkey's retina that was, say, 580 nanometers, which in isolation looks yellowish, but if you surrounded it with other wavelengths in just the right way, you could make that V4 neuron respond as though the patch were red or green or whatever color the scene context implied. The neuron wasn't reporting wavelength, it was reporting the result of a computation. It was reporting what color that surface would be in the real world once you factored out the lighting. Zeki argued that V4 was essentially the brain's color center, and while that turned out to be a bit of an oversimplification, color processing is distributed across several areas, as most things in the brain are, the core insight held up beautifully. V4 is doing something far more sophisticated than a spectrometer. It's doing color inference. And here's where it connects back to something we touched on earlier. Remember the opponent processing idea from Ewald Hering? Red versus green, blue versus yellow? That recoding that starts in the retina with the ganglion cells? Well, by the time those signals reach V4 and the surrounding areas, the opponent channels have been transformed again. The brain is now comparing color signals across large regions of the visual field, looking at ratios and contrasts between surfaces, essentially asking not, what wavelength is coming from this spot, but how does this spot compare to everything around it? That's what Land was getting at with Retinex theory, retina plus cortex working together. And it's why optical illusions involving color are so effective. You've probably seen those images online where two squares on a checkerboard look like completely different colors. One looks dark, one looks light. But when you cover up the surrounding context, they're physically identical. Same wavelength hitting your eye. But your brain, doing its contextual subtraction, its illumination estimation, assigns them different colors. That's V4 and its network doing exactly what they evolved to do. They're just being tricked by an artificial scene that violates the assumptions the system normally relies on. Now, here's something worth sitting with for a moment. When V4 is damaged, when someone has a stroke or a lesion that takes out this area, they can develop a condition called cerebral achromatopsia. And this is different from the color blindness you might be thinking of, the kind that runs in families where you're missing one of the cone types in your retina. That's a receptor problem. Cerebral achromatopsia is a processing problem. The cones are fine. The retina is fine. The information is being sent up the optic nerve perfectly well, but the brain can no longer construct color from it. People with this condition describe the world as looking like a black and white film. Everything is shades of gray. And what's haunting about it is that they often remember what color looked like. They know roses are red. They just can't see it anymore. Some patients report that it's not just the visual world that loses color. Their dreams become grayscale too. Their mental imagery drains of hue, which tells you something deep about where color lives. It's not in the light. It's not even in the cones. It's in the cortical computation. Take away the computation, and color vanishes from your entire experience, waking and dreaming alike. So let's pull back a little and think about what's happening in the ventral stream as a whole, because color is just one thread of what it's weaving. Remember, this is the what pathway, the one flowing down into the temporal lobe, And its job, its grand project, is object recognition, figuring out what you're looking at. And that sounds simple until you realize just how hard a problem it actually is. Think about what it means to recognize a face. You can recognize your best friend from the front, from the side, in dim light, in bright light, with a new haircut, wearing sunglasses, across a crowded room where their face is maybe 60 pixels wide, if you think of your retina like a camera sensor. You can recognize them in a photograph, in a caricature, even in a rough sketch. Your visual system is somehow extracting the essence of that face, the spatial relationships between features, the proportions, something abstract and invariant, and matching it against a stored representation. And it does this in a fraction of a second, usually without any conscious effort at all. You don't sit there analyzing nose width and eye spacing. You just look and know. That's the ventral stream at work, and it involves a cascade of areas beyond V4, regions in the inferior temporal cortex where neurons have increasingly complex preferences. This is where the hierarchy we talked about with Hubel and Wiesel really pays off. Back in V1, neurons respond to oriented edges, small, simple features. As you move forward through V2, V4, and into the temporal lobe, the receptive fields get larger and the preferred stimuli get more complex. In the inferior temporal cortex, researchers found neurons that respond to specific shapes, to combinations of features, and, here's the famous finding, some neurons that respond specifically to faces. Charles Gross at Princeton was one of the first to report this, back in 1972, and people were skeptical. A neuron for faces? It sounded too specific, too neat. But it held up. There are populations of neurons in a region called the fusiform face area, identified clearly in humans using brain imaging by Nancy Canwisher at MIT in the mid-1990s, that respond much more strongly to faces than to any other category of object. Not to a specific face usually, but to the general configuration of a face. 2 eyes above a nose above a mouth, that basic template. And when this area is damaged, you get prosopagnosia, face blindness. Oliver Sacks, who we mentioned earlier, actually had this condition himself. He wrote about it with his characteristic honesty and wonder. He could see perfectly well. He could describe the features of a face in front of him, the shape of the nose, the color of the eyes, but he couldn't assemble those features into a recognized identity. He would fail to recognize close colleagues, sometimes even his own reflection. The information was all there, but the binding, the recognition, the "oh, that's Sarah," that was gone. And what's remarkable is how specific the deficit can be. People with prosopagnosia can often still recognize objects, read words, navigate spaces. It's faces specifically that become unreadable, which tells you that the brain has dedicated specialized hardware to face recognition, separate from its general object recognition machinery. And that makes evolutionary sense, doesn't it? For a social primate, recognizing individuals, friend or foe, parent or stranger, is about as high stakes as visual recognition gets. Now, running in parallel with all of this ventral stream processing, the dorsal stream is doing its own equally vital work. We touched on this, the where and how pathway, flowing up into the parietal lobe, and one of its most critical functions is motion perception. Your ability to see things move, to track a ball flying through the air, to judge the speed of an approaching car, to notice a subtle movement in your peripheral vision that might be a threat. This is centered on area V5, also called MT, middle temporal area. And the story of what happens when it's damaged is one of the most striking in all of neuroscience. We mentioned Ziel's patient briefly before, but she deserves more of the story. Her name, when it was eventually published in case reports, was typically given as Patient LM. She was a woman in her early 40s living in Munich who suffered bilateral strokes on both sides of the brain, that damaged area. V5 specifically, while leaving most of her other visual areas intact. And what she experienced was achinotopsia, the inability to perceive motion. She could see the world, but it was frozen. She described pouring coffee as terrifying because the liquid in the cup appeared to be a static, frozen column. She couldn't see it rising. And then suddenly the cup was overflowing. She couldn't cross the street because she couldn't judge whether cars were approaching or how fast they were going. A car would be far away, and then suddenly it would be right next to her, like a series of photographs rather than a continuous scene. People talking to her were unsettling because their mouths didn't seem to move fluidly. Their lips would jump from one position to another. Think about what that tells you. Motion isn't something your eyes detect directly. Your retina captures a series of still images, snapshots, essentially, updated continuously. The perception of smooth, flowing motion is constructed by your brain, specifically by V5 and its connected areas, which compare successive frames and compute the direction and speed of movement. When that computation is gone, the snapshots are still arriving, but they're no longer stitched into a fluid stream. The world becomes a slideshow. And LM's case, published by Josef Ziel and his colleagues in 1983, was one of the clearest demonstrations that motion perception is a distinct, separable function of the brain, not just an automatic consequence of having eyes that work. So here you are, lying still, and your brain is running all of these streams simultaneously. The ventral pathway is identifying objects, recognizing faces, computing colors. The dorsal pathway is tracking motion, mapping spatial relationships, guiding your hand when you reach for a glass of water on the nightstand. And these streams aren't fully independent. They talk to each other constantly, sharing information through cross connections. But they can be damaged independently, which is how we know they're genuinely separate systems. And yet, despite all of this parallel processing, despite dozens of visual areas each doing their own specialized computation, your experience of seeing is unified. You don't perceive color separately from shape, separately from motion. You see a single, coherent, richly detailed world, a red ball rolling across a green lawn, one percept, seamless. How does the brain bind all of these separate computations back together into a single experience? This is called the binding problem, and it's one of the deepest unsolved questions in neuroscience. Some researchers have proposed that synchronized neural oscillations, brain waves at particular frequencies, especially in the gamma range, around 40 Hz, might be the mechanism. The idea is that neurons in different areas that are processing different features of the same object fire in synchrony, and that synchrony is what links them together into a unified percept. Wolfsinger and others proposed this in the late 1980s and 1990s, and there's some experimental support for it, but it remains hotly debated. Nobody's really nailed it down. And that's worth sitting with for a moment, because it tells you something profound about where we are in understanding vision. We can trace the path of a photon from the moment it hits your cornea all the way through the optic nerve, into the thalamus, across a dozen cortical areas. We can describe the receptive fields, the neurotransmitters, the timing of action potentials. And yet, the final step, the thing that turns all of that distributed computation into the seamless experience of seeing, remains genuinely mysterious. 40 Hz oscillations might be part of the story. Feedback connections from higher areas back down to lower ones might be part of it too. But the honest answer, as of right now, is that we don't fully know how binding works. And I think that's kind of beautiful, actually. That the thing you do most effortlessly, just opening your eyes and seeing a unified world, is the thing that stumps us the most. But let's not leave it there, because even though the binding problem is unsolved in the deepest philosophical sense, the practical neuroscience of how your visual system handles the real world has gotten extraordinarily sophisticated in the last few decades, and there are some genuinely wild things happening at the frontier. So let's talk about what modern vision science has figured out and where it connects to the technology and medicine that touches your life right now. Start with something you've probably heard of: artificial intelligence that can see. Computer vision. The whole field, in a very real sense, grew out of attempts to understand and replicate what your visual cortex does. And the connection isn't metaphorical. It's direct and literal. Back in the late 1950s and 1960s, when Hubel and Wiesel were mapping out those simple cells and complex cells in CAT-V1, showing that the visual cortex processes information in a hierarchy, edges first, then combinations of edges, then more complex shapes. They were laying the groundwork for something that wouldn't fully bloom for another 50 years. Because in 2012, a computer scientist named Alex Krzyzewski, working with Ilya Sutskever and Geoffrey Hinton at the University of Toronto, entered an image recognition competition called ImageNet with a system called AlexNet. And AlexNet was a convolutional neural network, a type of artificial intelligence architecture that is explicitly, deliberately modeled on the hierarchical structure of the visual cortex. The first layers detect edges and simple textures, just like V1 simple cells. The middle layers combine those into more complex features, corners, contours, parts of objects, like complex cells and the intermediate areas, and the deeper layers recognize whole objects, faces, scenes, just like the ventral stream. And AlexNet crushed the competition so dramatically, cutting the error rate nearly in half compared to the previous best, that it essentially launched the modern deep learning revolution. Every time your phone recognizes your face to unlock, every time a self-driving car identifies a pedestrian, every time a medical AI flags a suspicious spot on a retinal scan, it's running a descendant of that architecture, an architecture inspired by the wiring diagram of your own visual cortex. Here's what's really interesting, though. As these artificial systems got better and better, researchers started comparing them back to the brain, and they found striking parallels. If you show the same image to a deep neural network and to a monkey, while recording from its visual cortex, the patterns of activation in the middle layers of the network correlate remarkably well with the patterns in intermediate visual areas like V4. Not perfectly, but well enough to suggest that evolution and gradient descent, the algorithm that trains neural networks, converged on similar solutions to the same problem. Which makes sense if you think about it. The problem of extracting meaning from patterns of light is the same problem regardless of whether you're made of neurons or silicon, and there may be only so many efficient ways to solve it. But your brain does things that current AI still struggles with, and those differences are revealing. Take the issue of robustness. You can recognize a friend's face in harsh shadow, in fog, from an unusual angle, after they've gotten a dramatic haircut. Your visual system handles variation effortlessly. Current neural networks, despite their power, can be fooled by tiny perturbations that would be invisible to you. Change a few pixels in an image of a panda in just the right way, and the network confidently declares it's a gibbon. These are called adversarial examples, and they highlight a fundamental difference in how biological and artificial visual systems generalize from experience. Your brain isn't just matching pixel patterns, it's building something deeper, a robust, flexible model of what objects are, informed by years of experience, by context, by the physics of how light behaves in the real world. We're still figuring out how it does that so well. And speaking of the real world, let's come back to something closer to home, closer to your actual eyes. Because one of the most remarkable developments in modern vision science isn't about understanding the brain at all. It's about fixing the eye when it breaks. And the progress here, in the last 20 or 30 years, has been extraordinary. Think about what we covered earlier, how the cornea does most of the focusing, how the lens fine-tunes it, how the retina captures the image. When any of those components fails, vision degrades. And for most of human history, there wasn't much you could do about it beyond grinding a piece of glass and holding it in front of your face. Spectacles were invented in Italy, probably in the 1280s, and they were revolutionary, but they're still just external optics compensating for an imperfect eye. The real dream was always to fix the eye itself. LASIK, laser-assisted in situ keratomileusis, arrived in the 1990s and did something that would have seemed like pure science fiction a century earlier. The surgeon cuts a thin flap in the cornea, folds it back, and then uses an excimer laser to reshape the underlying corneal tissue with sub-micron precision, removing exactly the right amount of material to correct the refractive error. Then the flap is laid back down, and it heals in place. The whole procedure takes about 15 minutes per eye. And the reason it works so beautifully is precisely because the cornea is that transparent, avascular tissue we talked about way back at the beginning. No blood vessels to get in the way. A relatively simple and predictable structure that can be reshaped and will maintain its new geometry. Over 30 million LASIK procedures have been performed worldwide, and the satisfaction rates are remarkably high, north of 95% by most studies. It's one of the genuine success stories of modern medicine, and it's only possible because we understand corneal optics and wound healing at a very detailed level. But LASIK can't fix everything. It reshapes the cornea, so it corrects nearsightedness, farsightedness, and astigmatism, problems of focus. It can't help you if the retina itself is damaged. And retinal disease is where some of the most heartbreaking vision loss happens, because unlike the cornea, the retina is neural tissue, and neural tissue, as we discussed, doesn't regenerate easily. Age-related macular degeneration, AMD, is the leading cause of vision loss in people over 50 in the developed world. It attacks the macula, that tiny central region of the retina where your foveal cones are packed most densely, the part responsible for sharp, detailed central vision, the part you're using to read, to recognize faces, to see fine detail. There are two forms. The dry form involves a gradual breakdown of the RPE, those retinal pigment epithelium cells that service the photoreceptors, recycling their spent disc membranes. Without healthy RPE, the photoreceptors starve and die. The wet form is more aggressive. Abnormal blood vessels grow beneath the retina, leaking fluid and blood, distorting and destroying the macula rapidly. For decades, there was essentially nothing doctors could do for either form. You just went blind in the center of your vision, gradually or suddenly, and that was that. Then, in the mid-2000s, came anti-VEGF therapy. VEGF stands for vascular endothelial growth factor. It's a signaling protein that triggers the growth of new blood vessels. In wet AMD, there's too much VEGF being produced, driving those destructive abnormal vessels. Drugs like ranibizumab, approved in 2006, and later aflibercept, are injected directly into the vitreous humor. Yes, directly into your eye with a needle, which sounds terrifying but is actually quick and well tolerated, and they neutralize the VEGF, causing the abnormal vessels to regress. For the first time in history, doctors could not just slow wet AMD, but actually reverse vision loss in many patients. The clinical trials showed that about a third of patients gained significant visual acuity, and the vast majority stabilized. It was transformative. And the frontier keeps pushing. Gene therapy for inherited retinal diseases became reality in 2017 when the FDA approved Retina Neparvovec, sold as LUXTURNA, for a rare condition called Leber congenital amaurosis caused by mutations in the RPE65 gene. The treatment delivers a functional copy of the gene directly to the retinal cells using a viral vector. Children who were essentially blind from birth gained functional vision. Not perfect vision, not full restoration, but the ability to navigate, to see faces, to move through the world with their eyes instead of their hands. It was the first gene therapy approved for any inherited disease in the United States, and it happened in the eye because the eye has unique advantages for gene therapy. It's small, enclosed, partially immune privileged, and accessible. You can deliver the treatment precisely where it's needed. And then there's the even more ambitious frontier, retinal prosthetics. The idea of a bionic eye. If the photoreceptors are gone, but the ganglion cells and optic nerve are still intact, could you bypass the dead photoreceptors and stimulate the remaining retinal circuitry directly with electrical signals? The Argus II, developed by Second Sight Medical Products and approved in 2013, did exactly this. A tiny camera mounted on glasses captures the scene, a processor converts it to electrical patterns, and an array of 60 electrodes implanted on the retinal surface stimulates the ganglion cells. The resolution is crude, 60 electrodes versus over 1 million ganglion cells, so patients see rough patterns of light and dark rather than detailed images. But for someone who has been completely blind, perceiving the outline of a doorway, the edge of a table, the presence of a person standing in front of you, that matters enormously. The technology is still primitive compared to the biological system it's trying to replace. And Second Sight itself ran into financial difficulties, which is a whole complicated story about the economics of rare disease medical devices. But the principle has been proven. You can interface directly with the retinal circuit and produce visual percepts. And here's what's quietly astonishing about all of this. Every one of these interventions, LASIK, anti-VEGF injections, gene therapy, retinal implants, depends on the detailed understanding of visual system architecture that was built up painstakingly over centuries. Helmholtz measuring the lens, Ramon y Cajal drawing retinal neurons, Hubel and Wiesel mapping cortical columns, Kufler finding center surround receptive fields. Each discovery was a brick, and now the wall is high enough that we can actually stand on it and reach things that were previously out of grasp. But there's another side to modern vision science that's less about fixing broken eyes and more about understanding something even more fundamental: the relationship between what your eyes deliver to your brain and what you actually perceive. Because one of the biggest revelations of the last few decades is just how much of what you see is not actually out there in the world at all. It's constructed, fabricated, assembled inside your skull from fragments of data and a lifetime of assumptions, predictions, and educated guesses. And I don't mean that in some vague philosophical way. I mean it in a measurable, demonstrable, neuroscientific way. Your brain is not a camera. It never was. And the more we learn about vision, the more that simple truth keeps revealing deeper and stranger layers. Think about what your retina actually sends to the brain. We talked about this. About 1 million ganglion cell axons in each optic nerve, carrying compressed, heavily processed signals. Center surround receptive fields. On and off channels. Edges and contrasts, not raw pixel data. By the time visual information reaches your cortex, it's already been filtered, compared, and abstracted multiple times. But here's what makes it even more remarkable. That stream of information has gaps. Literal gaps. You've got a blind spot in each eye where the optic nerve exits, and there are no photoreceptors at all. You've got blood vessels casting shadows across your retina that you never see. Your sharpest vision, the foveal region we talked about way back at the beginning, covers only about 2 degrees of your visual field, roughly the width of your thumb held at arm's length. Outside that tiny sweet spot, your resolution drops off dramatically, and your eyes are constantly making rapid movements called saccades, jumping from fixation point to fixation point several times a second, and during each of those jumps, your visual processing is largely suppressed. It's called saccadic suppression, and it means that for a surprising fraction of your waking life, you're effectively not seeing anything at all. So if you add all of that up, the blind spots, the blood vessel shadows, the narrow fovea, the saccadic blanking, the raw feed from your eyes is patchy, intermittent, and low resolution across most of the visual field. And yet you experience a seamless, stable, richly detailed panoramic world. You look around a room and it feels continuous, complete, high definition from edge to edge. That experience is a construction. Your brain is filling in the gaps, smoothing over the interruptions, painting in detail that isn't actually there in the sensory signal. It's doing this so seamlessly that you have no awareness it's happening. You feel like you're seeing everything, but you're not. You're seeing a small amount and believing the rest. This idea, that perception, is an active construction rather than a passive recording, isn't new. Helmholtz himself talked about it in the 19th century. He called visual percepts unconscious inferences, the idea being that the brain uses prior experience and contextual clues to infer what's probably out there in the world, and then presents that inference to you as if it were direct observation. It was a brilliant insight, and it was largely ignored for decades because it was hard to test. But starting in the late 20th century, a framework emerged that formalized Helmholtz's intuition in a rigorous way, and it's become one of the most influential ideas in modern neuroscience. It's called predictive processing, or sometimes predictive coding. And the person most associated with bringing it into the mainstream is Carl Friston, a British neuroscientist at University College London. The basic idea goes like this. Your brain is constantly generating predictions about what it expects to see, hear, feel, and experience in the next moment. These predictions flow down from higher cortical areas to lower ones, from the abstract, conceptual levels of the hierarchy down toward the sensory surfaces. Meanwhile, actual sensory data flows up from the eyes, the ears, the skin. And what the brain really pays attention to is the mismatch between the prediction and the incoming signal. That mismatch is called a prediction error. When the prediction matches reality, the error signal is small and processing is efficient. The brain basically says, "Yep, everything's as expected. Carry on." When something unexpected happens, a flash of movement in your peripheral vision, a face where you expected a blank wall, a color that doesn't fit the scene. The prediction error is large, and that signal gets amplified and sent up the hierarchy for further processing. Your attention snaps to it. Your brain updates its model. Think about what that means for everyday seeing. Most of the time, you're moving through familiar environments: your home, your office, your neighborhood. Your brain has strong predictions about what's there. The chair is where it always is. The walls are the color they've always been. The coffee mug is on the desk. In those moments, your brain barely needs the sensory input at all. It's running mostly on its internal model, its prediction, and just checking the incoming data for errors. You're seeing your expectations as much as you're seeing the light. And this is why change blindness is such a striking phenomenon. Researchers like Daniel Simons and Christopher Chabris demonstrated in the late 1990s that people can fail to notice enormous changes in a visual scene. A person swapping out for a different person during a brief interruption, a gorilla walking through a basketball game, because if the brain's prediction doesn't include the change, and the change happens during a moment when the prediction error signal is suppressed or masked, it simply doesn't register. You look right at it and don't see it. Not because your eyes failed, but because your brain decided it wasn't there. And this is where those visual illusions we talked about earlier, Adelson's checker shadow illusion, the way your visual system assigns color based on context rather than raw wavelength, start to make even deeper sense. They're not bugs in the system. They're features of a prediction engine that's optimized, not for accuracy in the physicist's sense, but for usefulness in the biological sense. Your brain doesn't care what the exact luminance of that square on the checkerboard is. It cares whether that surface is light-colored or dark-colored, because that's the information that helps you recognize objects, navigate spaces, find food, spot predators. So it computes the most useful interpretation given the context, and that's what you see. The illusion only becomes apparent when someone strips away the context and shows you that two patches have the same physical luminance. In the real world, the brain's interpretation is almost always more useful than the raw data would be. Here's the thing, though. This constructive, predictive nature of vision means that what you see is shaped not just by the light entering your eyes, but by everything you know, everything you've experienced, everything you expect. Expertise changes perception. A radiologist looking at a chest X-ray literally sees things that an untrained person cannot. Not because their eyes are different, but because their brain has built a rich predictive model of what lung tissue should look like, and deviations from that model pop out as prediction errors. A birdwatcher spots a rare warbler in a hedgerow that you and I would walk right past, because their visual system is tuned to detect the specific pattern of movement, the particular shade of olive green, the shape of the silhouette. They're not just paying more attention. Their perceptual experience is genuinely different from yours. The construction is different because the priors are different. And emotion shapes it too. There's research showing that people who are afraid of spiders perceive spiders as larger than they actually are. People standing at the top of a hill with a heavy backpack judge the slope as steeper than people without the backpack. Your body's physiological state feeds into the predictive model and changes the percept itself. You don't just see the world and then react to it emotionally. The emotion is baked into the seeing. Now, you might be thinking, if so much of vision is constructed, predicted, filled in, how do we ever see anything accurately? And the answer is that we don't need to, not in the way a camera does. We need to see things usefully. Evolution didn't optimize the visual system for producing a faithful reproduction of the electromagnetic radiation bouncing around the environment. It optimized it for keeping you alive and helping you reproduce. And those are different goals. The visual system that survived is the one that's fast, efficient, and good enough. The one that spots the predator, recognizes the face, judges the distance to the branch you're about to grab. Precision in an absolute physical sense was never the point. And yet, and this is what I find quietly beautiful about the whole thing, despite all the construction, all the filling in, all the prediction and inference and shortcutting, the system works astonishingly well. You navigate complex three-dimensional environments at speed. You read facial expressions in a fraction of a second. You catch a ball thrown to you from 30 feet away, which requires your brain to calculate a trajectory, predict where the ball will be in the future, and coordinate your hand to arrive at that point in space at exactly the right moment. All in real time, all without conscious effort, all built from a patchy, intermittent, heavily compressed signal arriving through two small openings in the front of your skull. And I think that's the thread that ties everything together from the very beginning of this whole journey to right here at the end. The human eye and the visual system it's connected to. This isn't just an optical device. It's not just a biological camera. It's a profoundly integrated system that spans physics, chemistry, cellular biology, neural circuitry, and the deepest questions about consciousness and perception. It starts with photons hitting the tear film on the surface of your cornea, bending through that elegant curved surface, passing through the aqueous humor, being metered by the iris, focused by a living crystalline lens that changes shape on demand, traveling through the vitreous, landing on an inside-out retina where photons are caught by molecules of retinal nestled in opsin proteins, triggering a cascade that hyperpolarizes a photoreceptor, which modulates glutamate release onto bipolar cells, which feed into ganglion cells with center surround receptive fields, whose axons bundle into the optic nerve, partially cross at the chiasm, synapse in the LGN, project to V1 where simple cells detect edges and complex cells detect moving edges and hypercomplex cells detect corners, and then the signal fans out into dorsal and ventral streams, into V4 for color and V5 for motion, and the fusiform face area for faces, and somehow, all of that gets bound together into a single unified conscious experience of seeing. And we still don't fully understand that last step. The binding problem. The hard problem of consciousness. How does neural activity become experience? How does electrochemistry become the redness of red, the face of someone you love? The way late afternoon light looks falling through a window? That remains genuinely, honestly, beautifully mysterious. And I think there's something fitting about ending here with that mystery, because the human visual system is one of those things that gets more wondrous the more you understand it, not less. Every layer you peel back reveals another layer. Every answer opens new questions. So the next time you open your eyes in the morning and the world just appears, effortless, immediate, whole, maybe for a moment you'll remember some of what's actually happening behind that experience. The cornea bending light, the lens adjusting, the photoreceptors catching photons, the retinal circuits compressing and comparing, the cortical hierarchy building edges into shapes, into objects, into meaning, the predictions flowing down, the errors flowing up. Centuries of human curiosity, from Ibn al-Haytham's darkened room to Hubel and Wiesel's electrode in a cat's brain to a surgeon threading a gene therapy vector beneath a patient's retina. All of that knowledge, All of that effort, all of those lives spent figuring out one piece of the puzzle, just so we could begin to understand the thing you do without thinking every single moment of your waking life. You see, that's it. That's the miracle. And now you can close your eyes and let all of this settle and drift. The system that's been working so hard all day, catching photons, computing edges, building your world, it gets to rest now. The photoreceptors go quiet. The cortex softens its predictions, and you can just be here, in the dark, where seeing stops and sleep begins.